Welcome to this lecture on the physiological action of hormones. Have you ever wondered how a tiny chemical released in one part of your body can instantly make your heart race or help your muscles absorb energy? Today, we are going to uncover the fascinating biological and mathematical mechanisms that make these vital processes possible. To understand how this works, let us start with a simple analogy. Imagine you're listening to a radio station, broadcasting a signal across an entire city. Even though the radio waves are everywhere, only the radios specifically tuned to that exact frequency will play the music. In the human body, hormones act just like those widespread radio broadcasts. They travel everywhere through your bloodstream. However, their physiological action only occurs when they find a target cell equipped with the correct antenna, which we call a receptor. Once the hormone binds to this receptor, it changes the behavior of the cell. Now that we understand the radio analogy, let us look at what actually happens inside the cell. The hormone itself does not usually enter the cell to do the heavy lifting. Instead, it acts as a first messenger. By binding to the receptor, it simply flips a molecular switch on the cell surface. This single binding event initiates a massive cascade of internal signals, a process known as signal transduction. Depending on the specific hormone, this action can alter the permeability of the cell membrane, activate dormant enzymes, or even enter the nucleus to turn specific genes on or off. This mechanism we just explored leads us to a critical question. Why does this matter so much? The physiological action of hormones is the absolute foundation of homeostasis, which is your body's ability to maintain a stable internal environment. Without this precise action, your body could not regulate blood sugar after a heavy meal or respond to a sudden physical threat. For example, in a disease like diabetes, the hormone insulin is either missing or its action is blocked at the receptor level. Understanding this allows medical professionals to design life-saving drugs to treat endocrine disorders. Before we move on to the mathematics, let us visualize this process with a diagram. On the left, you can see a blood vessel with small, circular hormone molecules flowing through it. Some of these molecules exit the vessel and approach the target cell on the right. Notice the Y-shaped receptors embedded in the target cell membrane. They act like molecular locks, waiting for the right chemical key. When a hormone molecule successfully binds to one of these receptors, it triggers the physiological action or cellular response inside the cell, represented by the glowing gear in the center. Now that we have a clear visual understanding, we must recognize that to truly understand how hormones exert their effects, we need to look at the mathematics of receptor binding. The magnitude of the physiological action is directly proportional to the number of receptors that are currently occupied by the hormone. A cell will respond weakly if only a few receptors are bound, and strongly if many are bound. This means we need a way to calculate the fraction of occupied receptors to predict how strongly a cell will respond based on the amount of hormone present in the blood. This need to predict the cellular response brings us to a fundamental equation from biochemistry known as the fractional occupancy formula. Conceptually, this formula represents a competition between the amount of hormone available and the receptor's natural tendency to let go of the hormone. Let us break down the symbols. Y represents the fractional occupancy, or the percentage of receptors currently bound. H, in brackets, stands for the concentration of free hormones circulating in the blood. Finally,
KD represents the dissociation constant, which measures how tightly the receptor binds the hormone. If the hormone concentration increases significantly, the value of Y approaches 1, meaning maximum physiological action.
Now that we have our formula, let us see this with a concrete example involving insulin. We are given a target muscle cell with a known dissociation constant of 2 nanomolar. After a meal, the patient's circulating insulin concentration rises to exactly 2 nanomolar. We need to find the fraction of bound receptors. We choose the fractional occupancy formula because it directly relates these variables. We substitute the hormone concentration of 2 into the numerator and denominator, and the dissociation constant of 2 into the denominator. Dividing 2 by 4 gives us 0.5. Physically, this means exactly 50% of the insulin receptors are active. The result we just calculated reveals a very important rule in physiology and pharmacology. Let us prove why this happens universally, not just for insulin. We are setting out to derive the half maximal rule. We start by assuming a scenario where the circulating hormone concentration exactly equals the receptor's dissociation constant. Mathematically, we set the concentration H equal to KD. Next, we substitute KD in place of H in our fractional occupancy formula. This step is crucial because it allows us to see the inherent mathematical relationship when the concentration perfectly matches the receptor's affinity. Continuing with our derivation, we now simplify the equation. In the denominator, adding KD to KD gives us 2 times KD. We now have KD divided by 2KD. The KD terms in the numerator and denominator cancel each other out perfectly, leaving us with 1 divided by 2, or 0.5. We have just proven that whenever the hormone concentration equals the dissociation constant, exactly half of the receptors are activated. This produces a half-maximal physiologic response, a fundamental baseline used by medical professionals to understand drug and hormone dosages. Now that we understand the baseline, let us apply our formula to an extreme scenario, a massive hormone surge. Imagine an intense stress response where adrenaline spikes to 98 nanomolar. The heart muscle receptors have a dissociation constant of 2 nanomolar. We want to calculate the new fractional occupancy to understand the heart's reaction. We use the same formula, replacing the hormone concentration with 98 while the dissociation constant remains 2. Dividing 98 by 100 gives 0.98. Physically, 98% of receptors are bound, resulting in a rapidly racing heartbeat. We have covered a lot of ground today, so let us review our key takeaways. First, the physiological action of a hormone relies entirely on specific receptors acting as molecular locks. Second, the hormone acts as a first messenger to trigger a cascade of internal cellular responses. Finally, the magnitude of this response is tightly controlled by the hormone concentration and the receptor's affinity, as proven by our fractional occupancy calculations. You now understand the fundamental mechanisms that coordinate everything from your daily metabolism to extreme survival responses. Great job!